All right, everybody, welcome back to part 3 of muscle tissue. And so in this video, I'm going to cover these last 3 learning goals. So we're going to talk about whole muscle contraction, talk about how ATP is regenerated, and then finally describe muscle fatigue. So let's go ahead and get started. So essentially, whole muscle contraction, it's going to be similar in some ways to single fiber contraction, but it's going to differ a bit as well. So whole muscle contraction, the entire big picture purpose here is to produce muscle tension. And tension refers to the force that is going to be exerted on an object or a load in order to move that load. So let's think about our biceps muscle, for example. And let's do this. Okay. Here we have our arm. Okay. And essentially, our biceps muscle then is going to be located about right here. Okay, and so that would make this our elbow joint. So our biceps muscle, whenever it contracts, it has the job of bending our elbow. Okay, so the force that the biceps muscle exerts has to be enough force to move our forearm and our hand. Okay, so that's what I mean by muscle tension. The force and the duration that the muscle is contracting depends on different frequencies and different intensities of the motor unit stimulation, which remember in unit 2, in part 2, I should say, is when we talked about motor units. And so because the response of the muscle is variable and dependent on the frequencies and the intensities, we call the whole muscle contraction, we describe it as being a graded response. And whole muscle contraction, the muscle itself may or may not shorten. It really just depends on whether the contraction is isometric or isotonic. So I put this slide in here because I want to make a point that this is where we're going to differ from the individual fiber contraction. So in individual fibers, we have the all or nothing principle. And the all or nothing principle, if you remember, is when there is enough change in membrane potential to cause an action potential to propagate down the muscle fiber. All right, so basically this all or nothing principle says the individual fibers will either contract to their fullest, and the actin and the myosin will slide past each other and be fully contracted, or the individual fiber will not contract at all because there's not enough voltage change in the membrane potential. These graded potentials, however, refer to the whole muscle. And in whole muscle, some of the fibers are contracting, but not necessarily all the fibers are contracting at the same time, right? So it just depends on those motor units, because remember, those motor units so let's do this. Let me just draw something in here for you. If we are looking at the individual fibers, we had a picture kind of something like this, where we had one motor unit coming in and synapsing on maybe these 2 fibers. So this is motor unit 1. And then we had another unit coming in it's going to synapse maybe on this fiber back here, and then on this fiber back here. And this was motor unit 2. Now, as these individual fibers are located in the whole muscle, say we have stimulation occurring to motor unit 1. That means that only this fiber and this fiber are going to be shortening. Okay, these other 3 fibers are going to stay put, right? Because motor unit 2 was not being stimulated. And so the response of this muscle as a whole is going to depend on how many fibers are contracting or being stimulated simultaneously. All right, so in this instance, only these 2 fibers are going to be being stimulated, and therefore the muscle maybe will only contract a little bit. This is good though. This is what we want, because if we had every time our whole muscle being contracted, our movement would be very strong, powerful, and jerky every single time that we were stimulated. And that's not what you want, because you want to be able to do things like softly hold a pencil in your hand. This is great because if only 2 of the fibers are contracting, then we can do these more fine, small, precise movements. Contrarily, if we decide that we're going to climb a rope, then at this point, we would go ahead and stimulate both of them, both motor units. And now all of these fibers are going to shorten. And this will provide us with more strength in order to be able to tightly squeeze that rope when you're climbing it. Okay, so I just wanted to bring up this point that graded potentials and the all or nothing potential, they are related, but they are different entities. And so in muscle and whole muscle, we're going to be talking about graded potentials. All right, so again, whole muscle contraction depends on the motor units. Okay, and so this is really similar to the picture that I just drew. And each muscle is served by at least one motor nerve, and the motor nerve contains the axons of hundreds of these motor neurons. And the axons then are going to branch into terminals, which is going to form neuromuscular junctions with single fibers. So in this picture, what we're seeing then is here's a neuromuscular junction, here's a neuromuscular junction, here's a neuromuscular, here's a neuromuscular, here's a neuromuscular junction. So in this example, and then there would be hundreds of these, but in this example, it's 2 separate motor units that are synapsing on individual fibers, okay? The nerve muscle functional unit, so everything branching off of this purple motor neuron axon is going to be a motor unit, and the motor unit is the functional unit here. So it consists of one motor neuron, which we see here, and all of the muscle fibers that it's going to supply. And so motor unit 1 here is going to supply 2 fibers. Motor unit 2 over here is going to supply 3 fibers. All right. The smaller the number of fibers that the motor unit supplies, the better fine control that we're going to have of muscle. So since motor unit 1 is only stimulating 2 fibers, it's going to provide a more smooth and better fine control. So being able to lightly hold your pencil compared to motor unit 2, which is having to basically divvy up amongst 3 fibers. Okay, so it's going to cause 3 fibers to contract. Muscle fibers from a motor unit, they're spread out throughout the whole muscle, and so stimulation of a single motor unit is only going to cause weak contraction of the entire muscle. So you can see in this picture that motor unit 1, the 2 fibers that it is going to stimulate are not sitting right next to each other. All right, and this is good because we want them to be sort of spread out, so it has a better overall ability to affect the entire muscle. Okay. So let's talk a little bit more about these graded muscle responses. And I just want to point out that normal muscle contraction is usually very smooth, and the strength of the contraction is just going to depend on what you need to do with the muscle. And as we kind of roll through this here, again, the responses of the muscle as a whole vary the strength of the contraction for different demands. And this is required for proper control of your skeletal movements. The responses are graded by the frequency that the fibers are being stimulated and also the strength of the stimulation. And so we're going to go through each of these things over the next several slides. So in terms of frequency, it can either be a twitch, a wave summation, unfused tetanus, or fused tetanus. And we'll go through each of those things. And then in terms of the strength, we're going to talk about the recruitment and size principle components of the strength of the contraction. Now, before we get into twitch here, I just want to point out that a muscle twitch is only seen in a laboratory setting or when a person has neuromuscular problems. But normal muscle, you shouldn't have muscle twitches, okay? So I just wanted to point that out before I move on to the next slide and talk about muscle twitches. So when I say muscle twitch, I mean that a muscle twitch is just the simplest contraction that results from a muscle fiber's response to one single action potential, okay? So in this case, we're having only one action potential. The muscle fiber is going to contract quickly and then it's going to relax. And the twitch is going to be seen, it can be observed on what's called a myogram. And a myogram basically is just a line that records the contraction activity. So that's what you see over here. This is a myogram. Now, a twitch, let's go ahead and bring this out here. Okay, so starting over here at 0, we have one action potential come through, okay? Now, the strength and the duration of the twitch is going to be different in different muscles. And so it really just depends on the whole muscle's metabolic properties and the enzymes that are between the muscles. So for example, the eye muscle contraction is really quick and very short-lived, right? So eye muscle contraction is being shown here, this lateral rectus, okay? As we can see, the lateral rectus, the contraction is really short in terms of time. Okay. Now, time is located down here on this axis, x-axis. All right, so in terms of time, the eye muscle was pretty short. This is, again, just the result of one single action potential coming through. Whereas larger fleshy muscles like the calf muscles contract more slowly and they hold that contraction longer. So the gastroc muscle is one of the calf muscles. And we can see there is its it contracts that long. Okay. The soleus is yet another calf muscle, and it begins its contraction and lasts all the way up to 200 milliseconds. All right, and so what we're seeing then in this picture is just a comparison of different muscles and their durations. One thing to notice about this picture, however, is how much tension is here. If we notice that single action potential in terms of the strength that it gave the muscle, all three muscles were given the same amount of tension. Okay, so they resulted in the same amount of tension. They just held that contraction for different lengths of time. Okay. We're going to talk more about the twitch, so don't you worry. The muscle twitch itself occurs in 3 phases. So the first phase is the latent period. And the latent period is lining up. So what's happening during the latent period is these are the events that are coinciding with the excitation contraction coupling. Okay, so you don't see muscle tension yet, right? First, we have to go through excitation contraction coupling. The second phase is the contraction phase. And this phase is the phase that corresponds to when we're forming cross bridges, right? And so here's where you're starting to see muscle tension increasing. Finally, the third phase is the period of relaxation. And the period of relaxation is when calcium is re-entering the sarcoplasmic reticulum, and therefore tension is decreasing at a slow rate until it reaches 0. So as we can see based on this myogram over here, that muscle contracts faster than it relaxes. So you can see in the contraction phase, we spend only a relatively short amount of time in comparison with the relaxation phase where we spend most of the time. Now this bottom graph is showing you a myogram of 2 individual twitches, right? So if we have one twitch here, and then at the end of this period, we added a second action potential, this graph would repeat itself. So this one down here is showing you two individual twitches. But since these two stimuli are spread out far enough in terms of time, the muscle is given plenty of time to relax the entire way before it contracts again. And therefore, the tension that's created is going to be the same both times, right? Okay. Here's where that starts to differ. If two stimuli are received by a muscle in rapid succession, so here we have the first stimuli coming in, and so we'd have the latent period, and then we have the period of contraction, and now we're starting to relax, and then all of a sudden the muscle fiber is hit with another action potential. It's going to contract again because the muscle fiber didn't have time to completely relax before that second stimulus was added. And so what happens here is that extra calcium is going to be released from the sarcoplasmic reticulum due to that second stimulus coming in. And this is going to cause even further muscle shortening and thus resulting in an increased amount of tension. All right. So in these cases, temporal summation or wave summation is exactly what it sounds like. All right. So wave summation or temporal time. So wave summation, so we can see that in a normal situation, this would have, let's do a different color because this color is not really doing great for me. This would have come back down. All right, this entire wave would have come back down to rest. But because we were hit with the second stimulus here, instead of relaxing all the way, we start to build up this tension, and that's where the summation comes in, okay? And we are adding waves together because of this time factor, that there's not enough time to fully relax before we got hit with that second stimulus. And so therefore we added tension, okay? Now, if the stimulus frequency or how often the muscle is stimulated increases, then muscle tension will reach a near maximum. And this will produce smooth, continuous contractions that add up, right? So this is still kind of along with summation, but if further increases in that stimulus frequency occur, then the muscle will progress to a sustained quivering contraction, and this is called unfused tetanus, right? So now we're talking about the idea that there's even less time between stimuli now. And so our waves, they're adding up and they're adding up until they reach a point where they can't really add any more because they're at their maximum tension. So now they're gonna just quiver. So now in fused or complete tetanus, we have the stimuli frequency even shorter in terms of the time factors, though they're occurring more closely together. So now the stimuli frequency is increasing, and thus muscle will reach its maximum tension. And this is referred to as fuse tetanus because, as you can see, all of the contractions will fuse into one smooth, sustained contraction plateau. Now, prolonged contraction here will lead to muscle fatigue, and we'll talk about that as we move throughout this lecture. So now we've talked about the frequency of muscle contraction, and let's talk about how we can, or how we do, I guess, increase the strength. Now, if the stimulus gets stronger, then the muscle is going to send that stimulus out to more fibers. And this is going to lead to more precise control of muscle. And so in terms of recruitment, that's essentially we're recruiting more fibers in order to complete and sort of handle the stimulus. The types of stimuli that are involved in recruitment include subthreshold, threshold, and maximal stimulus. So in subthreshold stimulus, the stimulus that's received, it's not strong enough, so you actually don't see any contraction. And so I'm going to just draw this. Would be everything over in this region. Okay, this is all sub-threshold stimulus. Okay. From here to here, this is all sub-threshold stimulus. All right. So as you can see, in terms of the voltage of the stimulus, it's not very strong. So here we have voltage over here on this left axis. So over here on this y-axis, we have the voltage. Here the voltage, it's pretty low, all right? And so we have to reach a certain threshold, a certain voltage in order to actually see contraction. And in subthreshold, we have the threshold is not met. Therefore, the number of fibers or of motor units and fibers, for that matter, being stimulated would be 0, and therefore we don't see any muscle contraction. Now, during threshold stimulus, now we have a stimulus that's strong enough to cause the very first observable contraction. Okay, so let's draw that in here. The first observable contraction once we reach threshold would be this one. Okay, so now we have met our threshold. We see 2 fibers that were probably innervated by the same motor unit being stimulated, and we see our first contraction here on the myogram. Now, as the strength of the stimulus increases, so as the voltage increases, more and more fibers are going to be stimulated. So we have an even stronger stimulus over here at number 4. And now maybe we have 5 fibers being stimulated, which will result in a stronger contraction compared to our threshold stimulus, right? So we'll have more tension in the muscle. This will increase and increase until we reach what's called the maximal stimulus, okay? So maximal stimulus here. Okay, everything over here. As you can see from this point, once we hit this maximal stimulus, all of the fibers in the muscle are being stimulated and our maximum tension, so the muscle is as strong as it's going to be. And the important thing to note here is that even by increasing the strength of the stimulus, so even by increasing the voltage, see this is stepping up step by step here, so we're increasing the voltage, it doesn't result in any stronger contraction, right? So we have a stimulus that once we hit this maximal area and all of our fibers are contracted, we don't have any more fibers to contract, so we can't possibly have a stronger contraction. Okay. Now, this idea of recruitment that we just talked about is dependent and it works on what's called size principle. And this whole idea is just that motor units with the smallest muscle fibers are going to be recruited first, and motor units with larger and larger fibers are going to be recruited as the stimulus intensity increases. And the largest motor units are activated only for our most powerful contractions. Additionally, these motor units in the muscle, they usually are contracting asynchronously. And so this just means that some of the fibers are contracting while the other ones rest. And this helps to prevent fatigue. So this is just showing you that first, the small fiber motor units are recruited, and then as time goes on, then we'll recruit the mediums, and then we'll recruit these large fiber motor units. And this stepwise fashion will lead to an increase in tension. Okay, so now let's move on to talking about muscle tone. Muscle tone is the idea that muscle is in this constant, slightly contracted state all the time. And this is due to our spinal reflexes. And so we have groups of motor units that are alternately activated in response to input from stretch receptors that are in the muscles. And this helps to keep our muscles firm, healthy, and ready to respond. So, for example, the lifting of the arms will activate the core muscles so that we're able to stay in this seated position. So as we lift our arms, our abdominal muscles are activated to help steady our trunk. And we have back muscles that work to support the weight of the arms. We also have neck muscles that will be activated to help keep the head steady so that whenever we are lifting our arms, we don't fall forward or backwards or sideways. We are still able to maintain our posture. So this is the idea of muscle tone. So all of the muscles in our body are slightly contracted essentially at all times. At the beginning of this lecture, I pointed out that whole muscle contraction doesn't necessarily mean that the muscle has to shorten. The fibers, however, work on that all or nothing principle where if they're stimulated, they are going to shorten, but that does not mean that the whole muscle will shorten. The whole muscle shortening or not depends on what type of contraction we're going to be doing. So in isotonic contractions, we do change the length in order to move the load. So I put in this picture for you to kind of give you an idea of the two types of isotonic contraction. And so isotonic means same tone, same muscle tone. That means you're using the same amount of strength to move the load, but the length of the muscle is going to change. So in concentric contractions, the muscle is going to shorten. And the example I have here for you is you're going to start in this partially flexed elbow position, and you're going to do a bicep curl. You're going to lift those weights. This means that by performing this action, the biceps muscle is shortening. So this is a concentric isotonic contraction. However, the other type of isotonic contractions are these eccentric contractions. And the eccentric contraction would be starting out with the weights up by your shoulders and your elbow fully flexed, and slowly lowering them to your hip region in only a partial elbow flexion position. Okay, so in this case, our biceps muscle, as we lower our forearms, the biceps is going to be relaxed. It's going to be lengthening, but it's still generating that force because it's still supporting the weight that is in our arms here. Okay? So this is sort of a animated version of what's going on here. So in this one's showing you concentric isotonic contraction. And actually, I'm going to go back for 1 second because I want to make a point that even though, so in this isotonic contractions, remember I said that isotonic means same tone. And it's same tone because the load is staying the same. It requires the same amount of tension to move this load up as it does to move this load down. Okay, the only difference being that the length of the muscle itself is doing the changing in terms of its length, right? So when we move up, the muscle gets shorter, and when we move back down, the muscle gets longer. But the same amount of tension is required to perform both of these actions because the weight of the load is not changing. Okay, I just wanted to make that clear before I moved on. So again, this is that animated isotonic contraction here. So concentric, specifically concentric isotonic contraction. So when the muscle gets stimulated, so we have some sort of stimulation coming in. Okay. Then the muscle is going to develop enough force or enough tension to move the load. And the load down here is going to be 3 kilograms. So once enough tension is developed in this muscle, the muscle is going to shorten and it's going to move the load. And it's important to point out that before we move on here, Once enough tension is developed, and the very moment that we start lifting this load, we use the same amount of force for the rest of the shortening, okay? The same amount of force to move it this much off the ground as it is to move it this much off the ground. And that's kind of what we see over here in this graph. So once we have reached our peak tension, then the tension in that muscle is going to stay the same. The bottom graph is showing you the length over time. Okay, so if we start out at 100% length, right? So it starts out as long as it can be. Once we reach the peak tension, so the amount of force it would take to start lifting this load, then the muscle itself starts to get shorter until the muscle relaxes, and then it will get longer again. Okay. On the sort of flip side of things, the other type of muscle contraction is what's called isometric. And if you're hopefully guessing what that means, this means same, the iso, and metric means length. So this is contraction where the length of the muscle is not changing. This occurs because the load is greater than the amount of tension that the muscle can generate. So the muscle doesn't shorten or lengthen. This is kind of like trying to pick up a car. Or I'm going to just draw in a quick example of this is a wall. And here we are pushing against this wall. Okay, here's our little friend pushing against this wall. As we are pushing against the wall, our muscles are contracting because we're pushing as hard as we can. And so that's what we see over here. We are pushing as hard as we possibly can against this wall. But the wall is too heavy and it's not going to move because we're just not strong enough to move walls. All right. And so what you see is that even though we're pushing as hard as we can, we're using all of our strength to push against this wall, the length of our muscles is not actually changing, even though we are using all of our might. Okay. So that's kind of what this is going to show you as well. So isometric contraction, now where our muscles are attached to a weight that exceeds the muscle's strength, essentially. So it exceeds the peak tension developing capability. So even when this muscle is stimulated, the muscle can't shorten because it's not strong enough to lift the load. So the muscle just stays the same length. Okay. Now, in isotonic and isometric contractions, the electrochemical and the mechanical events are exactly the same, but the results are different. So in isotonic contractions, isotonic meaning same tone, So this is the one where the muscle can either isotonic is muscle shorter or longer. In isotonic contractions, the actin filaments shorten and cause the movements. However, in isometric, so this one's same length, like pushing up against a wall. The cross bridges are generating force, but the actin filaments do not shorten. So the myosin heads are spinning their wheels on the same actin binding site. Okay? So now we're going to start talking about muscle fiber types. So not all muscle fibers are equal. We have three different types of muscle fibers. We have slow oxidative fibers, which are built for long-term endurance type activities like distance running. And these slow oxidative fibers, they contract very slowly. The myosin ATPase activity, so the cross-bridge cycling is a slower activity. They have a lot of myoglobin, so this enables them to have a lot of oxygen present. They don't have a lot of glycogen. These slow oxidative fibers are the first ones that are recruited. All right, and they fatigue the slowest. They are very fatigue resistant, which, as you can imagine, would be ideal for endurance type activities like marathon running or something simple like maintaining posture. In terms of their structural characteristics, the slow oxidative fibers are very small. They have a lot of mitochondria and a lot of capillaries, and they appear red. Okay. So these slow oxidative fibers are the first class. Next, we have these fast oxidative fibers. And fast oxidative fibers, they contract really quickly. They have a lot of myoglobin, which is great, which means they have a lot of oxygen. They are recruited second. Okay, these are the second recruits. When we were talking about the recruitment and the size principle, these ones are recruited second. And they can fatigue. They do fatigue more quickly than our slow oxidative fibers, but not as quickly as our fast glycolytic fibers. And so these fibers would be best for short-term rapid movements like sprinting or walking. These fibers are relatively large. They're the largest of the three fiber types. They have many mitochondria and many capillaries, and their appearance is they're this red pinkish color. Okay. Finally, we have the fast glycolytic fibers. And fast glycolytic fibers are fast contraction, so fast myosin ATPase activity, but they don't have much myoglobin, which means they don't have much oxygen, which is why they're going to be called anaerobic. They are the third to be recruited, and they fatigue the most quickly out of the three types. So they're best suited for things that will require short-term, rapid, intense movements, like explosive strengths, for example, hitting a baseball or moving furniture. They are the medium-sized fibers. They don't have much mitochondria, which is why they run out of energy pretty quickly, and they appear white to pale in color. So we're going to talk more about this as we move on in a little bit. So here's that same table that I just had on the last slide. I just blew it up for you so you could see it a little bit better. All right, so now that we've talked about whole muscle contraction, I just introduced you to the idea that muscle fibers are not created equal. We have three different types, and the types that you have just depend on the activities that you do. And so now we're going to talk about the ways that ATP is regenerated during muscle contraction. So let's go ahead and get started with that. So I just said we're going to talk about ATP, but why is ATP important? Well, ATP supplies the energy that's needed for the muscle fiber to remove itself and detach the cross bridges. ATP is also needed to pump calcium back into the sarcoplasmic reticulum, and ATP is needed to pump sodium out of and potassium back into the cell after excitation contraction coupling. So if you remember back to those videos of specifically if you think about the cross-bridge cycle where you have ADP and inorganic phosphate attached to the myosin head and then released from the myosin head, and then ATP comes in and gets broken down, which provides you energy for the next cross-bridge cycle. Also, if you remember back to I think it was on the second part of this lecture, I drew for you the sodium potassium pump that after we depolarize, we have and repolarize, the sodium potassium pump helps to maintain our resting membrane potential. So whenever we have too much sodium inside of the cell, it pumps it back out, and then the potassium that just left the cell, it's going to bring back in. And the available stores of ATP are depleted usually within 4 to 6 seconds. So ATP is so important because it is the only source of energy for contractile activities. For this reason, we need to be able to regenerate ATP very quickly. Now, in muscle contraction, ATP is broken down into ADP and inorganic phosphate by the action of something called ATPase. And just to kind of give you some context here, the ACE part of ATPase, anytime you see ACE, this usually means it's an enzyme that breaks stuff down. So we had acetylcholinesterase. right? So esterase, acetylcholine esterase ends in ASE, and that was the enzyme that breaks acetylcholine down and removes it from the synaptic cleft. So anytime you see ACE, it's usually meaning an enzyme that's going to break something down. So ATPase is going to break ATP into ADP, and inorganic phosphate. So we have ATP up here. So ATP stands for adenosine triphosphate. So here's adenosine, the adenine plus the ribose makes adenosine. The triphosphate means there's 3 phosphate groups. Now, this is a very high energy bond, so this is where we get the energy from. And whenever this bond is broken, so whenever we use ATP, this bond here is the one that gets broken down, and we are left with ADP, which is adenosine diphosphate, 2 phosphate groups, 1 inorganic phosphate group, and that bond that we just broke is going to release energy. So how much ATP do we need do we need to keep muscles working anyways? Well, I wrote out this little scenario here. So there's about 300 myosin molecules per thick filament. And each of those myosin molecules will be located on multiple different myosin heads here. And we have to multiply the number of myosin heads by all of the sarcomeres that are in one myofibril. And then we have to multiply all of those sarcomeres on one myofibril by all of the myofibrils that are in one muscle fiber. And then we have to multiply one muscle fiber by all the muscle fibers that are in one skeletal muscle. And then we have to multiply that by all the skeletal muscles that are in the body. So therefore, this comes to the conclusion that a ton of ATP, literally can't even calculate it, is needed to keep muscles working. Okay, so this is why ATP is so important and why I spend time here talking about it. ATP is regenerated by 3 different mechanisms. So we're going to talk about the direct phosphorylation of ADP by creatine phosphate, an aerobic pathway, and an anaerobic pathway. So in direct phosphorylation of ADP by creatine phosphate, this creatine phosphate molecule is located in muscle fibers, and creatine phosphate donates one of its phosphate groups to ADP to instantly form ATP. Okay, so you have creatine phosphate, the CP over here, is going to donate its phosphate to ADP. So when ADP receives that phosphate, now ADP becomes ATP. And the enzyme that takes the phosphate from the creatine phosphate and adds it to ADP is called creatine kinase. In direct phosphorylation, muscle fibers are given enough ATP and creatine phosphate reserves to power the cell for about 15 seconds. All right, so this provides us with about 15 seconds worth of energy. And notice, we do not have to use oxygen for this method. Okay, no oxygen required. Aerobic respiration is the type that produces 95% of the ATP that we use during rest and during light to moderate exercise. This one requires oxygen and occurs in mitochondria. So in the aerobic pathway or aerobic respiration, first what happens is this thing called glycolysis occurs, and glycolysis just takes glucose that was stored in muscles, and glucose that's stored in muscles is called glycogen, and it breaks glycogen down into 2 pyruvic acid molecules. Okay, so we take glucose from glycogen, break down, and we break that down and we get pyruvic acid. And then that pyruvic acid is going to enter the mitochondria, where a series of chemical reactions is going to occur, and the end result is going to be carbon dioxide, which is going to be a byproduct, water, which is going to be a byproduct, and we are going to get 32 ATP molecules. This is going to provide hours worth of energy, okay? So aerobic respiration is the main source of ATP generation during rest and light to moderate exercise. If I go back just 1 second, notice here we have other things like fatty acids and amino acids going in here. Let's talk about that. So normally we break down glycogen and go through aerobic respiration this way. So we use our sugars first. But if we run out of glycogen to use for aerobic respiration, then ATP is generated using other sources. And so first thing we use is glycogen, right? And that's located in muscle. And we use that first to make ATP by aerobic respiration. If we run out of glycogen, then we will use glucose that's circulating in the blood. So if you've ever measured your blood sugar, you know that there is a certain amount of sugar in your circulating in your blood, and that sugar happens to be glucose. You're measuring your blood glucose levels. So if we run out of glycogen, then we start using glucose. If we're using this glucose, as you can imagine, this is going to drop your blood sugar. All right, and so you might become sort of lightheaded, dizzy, faint, that sort of situation. But at least we can make ATP for muscle use, right? So this will keep our respiratory muscles working and our cardiac muscles working and so on. If we run out of blood glucose to use, we can only take so much glucose from the blood before we have to have a certain amount of blood glucose. And so then what's going to happen is we're going to go ahead and switch our energy source to fatty acids. And basically this involves us taking these fatty acids from adipose tissue and using the fatty acids as the source for ATP production. So the fatty acids are going to enter the mitochondria and go through aerobic respiration. So this is our third source. And this one, we start using fatty acids after about 30 minutes of exercise. And so this comes into play if you're trying to, for example, lose weight, and you start exercising. In order to get to the point of burning fat, you have to exercise for at least for more than 30 minutes, because you don't actually start burning fat until you've reached that 30-minute mark. Okay, so that's just something to keep in mind. Finally, if we have no more fat to use, we have no more glycogen stores, we've pulled all the glucose we can out of the blood, then we switch over to using amino acids. And amino acids are the building blocks for proteins. And so essentially what we're going to do is we're going to start breaking down proteins. This is not ideal, breaking down proteins to use as an energy source, but we will use it as a last resort if we have to. So the third way that we can generate ATP is through what's called anaerobic respiration. And just to give you context here in terms of terminology here, anaerobic means no oxygen, okay? No oxygen required. Now, anaerobic respiration only yields like 5% as much ATP as aerobic respiration, but the plus side is that it makes ATP at a rate that's 2.5 times faster than aerobic respiration. So anaerobic respiration is used when oxygen isn't available, such as whenever you're doing high intensity activities. So what happens in the anaerobic pathway is first we go through glycolysis, just like we did with aerobic respiration. And so we end up with this pyruvic acid molecule. And from glycolysis, we got 2 ATPs. Now, in anaerobic respiration, if we don't have oxygen, then instead of sending the pyruvic acid to the mitochondria, what happens is that pyruvic acid gets converted into lactic acid. And lactic acid will then diffuse into the bloodstream and can be used as fuel by the liver, the kidneys, and the heart. And in the liver, the kidneys, the heart, those organs can actually convert lactic acid back into pyruvic acid, or in the liver, we can convert that lactic acid back into glucose. Now, I think it's important to point out here too that this pathway only provides maybe 30 to 40 seconds of energy. And in these sort of high intensity power activities, say you're like lifting weights or something, you can get muscle soreness from this or muscle cramps from this if you have too much lactic acid buildup. Okay, so that can be the source of muscle soreness or muscle cramps, is just the lactic acid buildup. So you're, I don't know, power cleaning and then all of a sudden you start cramping. This is usually because too much lactic acid is being built up. So I'm going to give this in a context of different exercises. So in the short duration, high intensity exercise, ATP that is stored in muscle is used first, and this is going to last about 6 seconds. And from the start of exercise to about the point where we reach about 16 seconds, that second 10 seconds, ATP is formed from creatine phosphate and ADP, so this direct phosphorylation. In this short duration high intensity exercise, from 30 to 40 seconds to the end of the exercise, glycogen that's stored in muscles is going to be broken down into glucose, which is oxidized to generate ATP. So this short duration high intensity exercise is when we're using anaerobic, anaerobic pathway, no oxygen pathway. Short duration, high intensity, like sprinting, for example. At the end of the sprint, your muscles feel completely exhausted because at this point you're going to do anaerobic pathway. Now, if you're doing prolonged duration exercise, for example, like marathon running or long distance running, then this is when ATP is generated by breaking down several different nutrient energy fuels. So this is the point at which we're talking about using our glycogen and using our fats, our fatty acids. We're also talking about using proteins if necessary. And this provides hours worth of energy. So this over here is the aerobic pathway, okay? This is for prolonged exercise. Okay, so here is the last learning goal that we're going to go through for the muscle tissue lecture. So we are finally approaching the end, and I know this was a long lecture, so thank you for bearing with me. But this is full of a ton of really good need to know information for class. All right, so earlier I mentioned about muscle fatigue, and so here we're going to talk a little bit about it even more. So fatigue is this physiologic inability to contract the muscle despite continued stimulation. So once the muscles become tired, it doesn't matter if they're stimulated, they won't contract, they're just too tired. Some of the causes of muscle fatigue can be things like ionic imbalances. So our potassium, our sodium, our calcium levels can change, and this would disrupt the membrane potential of the muscle fibers. Another cause of muscle fatigue would be an increased direct phosphorylation, so increased amount of organic phosphate from creatine phosphate and ATP breakdown. And this might interfere with the calcium release from the sarcoplasmic reticulum, or it could hamper the power. Another reason you can get muscle fatigue is if you have decreased amount of ATP. So as ATP levels drop, magnesium levels increase, and the increase in magnesium can actually interfere with the voltage sensitive T-tubule proteins. And then finally, the fourth cause here is you just have decreased glycogen, okay? And remember, glycogen is glucose stored in muscles. So if you have decreased glycogen, then you don't have the energy for your muscles to contract. The lack of ATP itself is rarely a reason for fatigue, except in very severely stressed muscles. And so what this graph over here is showing you that for isometric contractions, for example, so same length contractions, pushing up against the wall, if you give a whole bunch of stimuli to these muscles, eventually you're going to get tired of pushing against that wall. Your body is physically tired, and you start to decrease the tension of your muscles. If you have ever done a wall sit, for example, okay, up against the wall, knees at 90-degree angle, and you're sitting there, and eventually, after some certain amount of time, your legs are on fire, and your legs, they feel completely exhausted, and you have to stop doing the wall sit, okay? This is because your muscles are becoming fatigued. And so in this isometric tension, so you're doing the wall sit, your muscles, they have reached their tetanus, so they're just staying contracted because that's what's holding you at that 90-degree knee angle sitting on the wall. And then they become tired. And so the tension starts to decrease and decrease and decrease. And then maybe you stand up for a second, and then all of a sudden you go back down into that wall sit. Okay, so maybe you rest for 20 seconds and then you go back down into that wall sit. Well, the second wall sit, you're not going to be able to hold as long because your muscles are already exhausted. And so you see fatigue a lot more quickly in that second wall sit. Muscle fatigue can also be caused by lactic acid buildup. So in order to return to its pre-exercised state, then muscle oxygen reserves have to be replenished. And lactic acid needs to be reconverted to pyruvic acid. And the glycogen stores need to be replaced. And the ATP and the creatine phosphate reserves need to be resynthesized. So when we have a high respiration rate and a high heart rate after really strenuous exercise, we call this excess post-exercise oxygen consumption. So this excess post-exercise oxygen consumption occurs because this is referring to the extra oxygen that the body's going to need following exercise in order to replenish the oxygen on myoglobin, in order to reconvert the lactic acid to pyruvic acid, in order to replace stored glycogen, and to restore ATP and creatine phosphate reserves. Okay, so this EPOC occurs, your high respiration rate, you're essentially consuming more oxygen after strenuous exercise because you're going to need that oxygen to recover the muscle. And that concludes the end of our lecture. So I will leave it at that. And if you need me, again, I'll be on Zoom from 9:30 to 10:45 on Thursday.